All right, thank you. So actually, Bjarne, would you like to start with tell us who you are?
Okay, I'm an adult neurologist from the beginning, but then some peculiar patients caught my interest, and that is more than 40 years ago. And these patients were the first ever titinopathies and titinopathy patients identified, and 25 years before we found the gene. So that was the start of my sort of research interests, and those then, of course, expanded in our research lab in Finland, Helsinki, to include all of genetics, muscle tissue issues, everything regarding muscle diseases.
So I've referred to you to other people as the grandfather of Titin myopathies.
Okay, you see, I am a grandfather also.
I know you are, right? Mathias.
Right. Hi, very nice to see all of you. I'm Mathias Gautel. I am a BHF chair of molecular cardiology, which make very little sense to anyone, including me. I really trained as a medical doctor, but I became interested more in the molecular mechanisms of muscle structure, function, and assembly. Did a stint at the European Molecular Biology Laboratory in structural biology, and I'm now heading the School of Basic and Medical Biosciences at King's College in London, where we're interested in genetic diseases, structural biology, developmental biology, and biophysics of muscle. And we deploy quite a wide range of methods to understand the molecular impact of mutations in genes, including Titin, and how we may use that sort of analysis in interpreting genetic constellations.
Thank you. Anna.
Hello, everyone. I'm Anna Sarkozy. I'm a clinical geneticist by training, but working as a neuromuscular doctor at the Great Ormond Street Hospital in London with Professor Muntoni. We are the reference center in the UK for congenital myopathies and muscular dystrophies. And in this role, we are referred for diagnosis and for clinical assessment and management patients with titinopathies, mostly children. But in another life, I also have been involved with care of adult patients with adult onset titinopathies. Very, very glad to be here today.
Sandra?
Hi, my name is Sandra Donkervoort. I'm the genetic counselor working with Dr. Karsten Bodeman at the NIH in Bethesda, Maryland. We are heavily involved in seeing patients with rare unknown neuromuscular disease to get to an underlying genetic diagnosis. So we see a lot of the early onset Titin patients. We mostly see children, but also see adults here and there, and then are also involved in the more research-based, trying to find missing or hidden genetic variants to confirm diagnoses.
Thank you. So, Sarah, I guess we're moderating on the fly here, but I understand that each of our speakers has a brief presentation to make.
Right.
Make some of the community-generated questions.
Yeah, so I did collect the questions and sent them to the speakers, and they are going to incorporate the answers into them. And then if people have questions, if they can put them into the chat, and we can revert back to them at the end. So if we go ahead and turn it over to Dr. Gautel. And if you're not speaking, you can put your camera off if you like.
Right. I'm not sure I can get the screen share to work, but now it does seem to work. Okay, good. Right, so I'm going to tell you a little bit about the molecular biology of Titin and its functions, and try to address some of these questions, which, however, I believe are probably better answered by Bjarne and Anna.
I will get back to them, yes.
Yes, so I think as all of you will know, muscle contains these very regularly arranged structures, which are called sarcomeres, and sarcomeres essentially are the smallest contractile units of muscle containing actin and myosin. But they are so incredibly regular when you look at them in an electron microscope that for many decades, people have wondered how such a regular assembly of millions of protein subunits, all of which are ignorant to their neighbors, can take place. And if this regular arrangement is disrupted, we know that this can lead to very severe hereditary muscle diseases. So over the last 20, 30 years, it's become clear that one particular player is important in that regular assembly. It's this giant protein called titin, which is the biggest protein in your body. And this very giant protein is so big that it can span over roughly 1.5 micrometers, from a so-called Z disk to the so-called M band, which is the lateral edge of a sarcomere and the center of the sarcomere. So this is a really gigantic protein, which is common to both skeletal and cardiac muscles. So depending on where a mutation is, it can affect both of these muscle types or only one of them. So what is really big in terms of molecular terms? So if you think of a person and then a very big airplane like an A380 that can carry very many persons, then this molecule is indeed the Titanic, the biggest protein in the human body, and encoded by a very large gene. This gene has over 364 exons, encoding 169 immunoglobulin-like domains, 132 fibronectin-like domains, a kinase, and lots of other things that I refer to as stuff for the sake of simplicity. So this is a really complex gene. It's of course, genetically speaking, it's a very big target, and a very big target eventually will be hit. So over the last years, we're trying to disentangle these very many hits. And despite its very big size and complexity, and this is a understandably very mind-boggling slide, but all it shows you is that despite its big size, titin can be broken down in small, roughly 100 amino acid big domains, and these domains can be separated out by molecular biology tools and studied separately to understand both their function, for instance, their interactions with other sarcomeric proteins, or their stability or biophysical properties when they're hit by mutations. So we can cut this into amenable little subunits that we can understand. So it's a bit like a Lego machine that has lots of interacting little modules, and they are put together into a very big assembly. And that very big assembly of titin is not only big, but it's modular, it's multifunctional, it's dynamic, and it's elastic. So that summarizes titin. But anything that is big can also fail, and titin is certainly not too big to fail, quite the contrary. And over the last decades, and really starting with the seminal work by Bjarne, many disease entities have been linked to mutations in the titin gene. I'm not going to go into much detail here, and many questions still arise about the contributions of some of these putative disease-causing mutations. So how can we really tell them apart? Probably one thing that clarified the role of titin mutations more generally was the identification by the Zeitman labs of truncating mutations in the titin gene as a major cause of dilated cardiomyopathy. However, in this study, about 3% of unaffected persons carried truncating mutations, and that is far too common for such a relatively rare cardiac disease. So what's going on here? It's clear that truncating mutations become manifest by second hits, and the second hits currently debated are things like pregnancy, exercise, alcohol, or chemotherapy. However, there are also, of course, other mutations in titin than truncating mutations. And this is a slide I took in 2016. I've stopped counting since then, because there were 94,833 missense mutations, so amino acid exchanges. So how do we make sense out of that enormous number of variants? So the 1,000 genomes, of course, are just a glimpse on human evolution. They contain good, bad, and ugly mutations. But how can we discriminate the good, the bad, and the ugly? And that's what we've tried recently in a paper that just came out this year with very many collaborators, and Anna Sarkozy amongst them. And it is an interesting study, we believe, because it is the first time that we've systematically been able to look at a very large number of mutations. Sorry, this will be illegible on your screens, but you can look at the paper and contact me if you've got any questions. So we've looked at very many missense mutations co-inherited in all of these patients with a truncating mutation. And we've used a new web tool that we've developed together with Franca Fraternali's team at King's College, which you can look up on the internet in this URL here. And this helps on a structure basis to assess the potential impact of missense mutations in Titin. But we still need more clarity, and we're still trying to refine this tool into something that essentially gives you, on the click of a button, a yes/no answer, pathogenic or not. And that is still very challenging indeed, because when we look at just very common variants with mean allele frequencies of 0.49 or 0.2, so far too common to be pathogenic. Very common disease predictors give you all kinds of random results. But if you then look at a key characteristic of mutant domains, which is their destabilization, then there's no impact, or indeed one of these is actually more stable than the wild type. So clearly, very commonly used predictors like polyphen, SIFT, or ProVN are totally useless for the classification of missense mutations in Titin. So what we've done is we've looked at these structure-based predictions from TitinDB, picked the ones which were suspicious looking based on their structural positions, and then measured their actual physical properties by measuring their melting temperatures, so the temperature at which the domains fall apart. And we found that all of the myopathy-associated variants reduce the thermal stability by at least 17 degrees Celsius, so that's a fairly precise number. And some of these variants were so misfolded that they couldn't be even refolded. We've now got a panel that essentially covers currently all of the Titin A-band domains, and we can measure all of the stabilities and then measure the impact of disease variants on those panel domains. And when we do that also for more common variants in gnome or hypertrophic cardiomyopathy, we can see that a subset of these mutations are similarly destabilized as those mutations that are associated with the pediatric early onset myopathy. So there may be more destabilizing recessive mutations out there than we have thought previously. So to summarize, I think what we have learned is that Titin is a major and irreplaceable scaffold for the sarcomere and a major gene for hereditary myopathies. And truncating as well as destabilizing missense variants are really very frequent in the normal population. But how do we discriminate the bad from the neutral and the good? What we've shown is that destabilizing mutations can be both recessive or dominant, and some recessive variants can cause disease in a compound setting when they are expressed with recessive truncating variants. So indeed, recessive missense variants in Titin can be a second genetic hit that leads to the phenotypic penetrance of a truncation. That's what we think. And an accurate measurement and assessment of the missense variant is required, and that needs to include structural and functional data at the moment. And one of the steps toward this is this database that we've created to assess a structure-based impact. So I'd just like to thank my team at King's College and all of my collaborators. This is very brief, and of course, very crucially, all the clinical collaborators worldwide that we've worked together with. In fact, I thank you all for listening and stop sharing. I've been a bit over the 10 minutes. I seriously apologize.
We'll have to be.
I will be shorter, so no problem.
Why don't we hear from Bjarne next then?
Okay. Thank you for the invitation, and I will try to go more into sort of almost trivial issues. So I will go over the questions that were already posted to us to try to dissect and see what we can get out of them. I do not actually have any presentation. I have it on presentation, but I think I will just tell what I'm thinking. So one question was regarding exon skipping and other gene therapies for Titin. This is, of course, now at this stage, not an easy issue, and nothing much is currently directly available. But of course, we and others have been thinking of this. And just theoretically, Titin is not the worst gene to work on, for example, for exon skipping, because many of the exons are so-called in frame. So if you take out one exon, it does not disrupt the whole gene and protein product, but just takes out a piece. So that theoretically may give us opportunities. And depending on where that exon lies along the sarcomere, as Mathias Gautel showed, there are different parts of the big protein that tolerate such minor sort of losses of one exon better than others. And this could be maybe used, but these are at the moment speculations. We actually have one project ongoing, and that is related to the fact that Mathias Gautel also touched upon, that these so-called truncating mutations, so the severely disruptive mutations, most of them do not end up with any producing any protein at all. So in individuals with just one such mutation on one chromosome, the more or less normal chromosome, Titin on the other chromosome from the other parent is capable of producing totally enough of good working Titin. And that gave us the idea that in the dominant diseases, there are few dominantly inherited titinopathies, which means that if you have a mutation on just one chromosome coming from an affected parent, you will have the same, more or less the same disease. In these cases, at least theoretically again, you could take out the whole mutant chromosome allele. And the patient would probably produce well enough of normal or good protein from the other chromosome. So we have a project ongoing to do that in mouse models. Difference between Titin-related myopathy, dystrophy, limb-girdle muscular dystrophy, or titinopathy. Okay, these are terms. So with myopathy, we mean all types of muscle diseases. Can be inflammatory, can be even toxic or whatever. So every time something goes wrong in a muscle, it is a muscle disease or myopathy. Now, dystrophies are always genetic. So they are the subcategory of myopathies which have a genetic background. But in old days, when the diseases were named and discovered and reported, we didn't know exactly which was sort of genetic or not. So there is a lot of mixture of terms, but this is the fact that with the muscular dystrophy, we mean a genetically based disease, and is usually used when there is a progressive loss of muscle tissue, so muscle wasting over time. The limb-girdle muscular dystrophies is a subcategory of these muscular dystrophies, and used when the muscle wasting and weakness starts in the girdle muscles, as opposite to the distal dystrophies or distal myopathies, when weakness starts in the hands or in the feet. And titinopathy is again a subcategory of the genetically based muscle diseases. When titinopathies were reported based on clinical findings before we knew the gene, different terms were used. So some titinopathies are called myopathies and some dystrophies, but essentially that has just historical background. Titinopathies have mutations in the titin gene to cause the disease. So one question is, if I have a known pathogenic variant or variants in titin, meaning genetic confirmation, what value is it to get the muscle biopsy? Now, this relates now to the certainty of the confirmation, because I know that not all patients who have received a titinopathy diagnosis are fully clarified to have a titinopathy. So if the pathogenic variants identified in you have previously been well described and characterized, and we know they are pathogenic, then of course it is justified to make the final diagnosis. And then in those cases, a muscle biopsy is not needed. If we are talking about just one variant as the cause of the disease, it means again the subcategory of dominant titinopathies. And currently we actually have certainty of just two different types. One is the H-Murph disease, titinopathy, which can start with respiratory failure in adulthood and progresses to include also all limb muscles. And the other one is the one I worked with originally, the mild late onset distal myopathy, which causes the ankle weakness, dorsiflexion weakness, and foot drop at late ages. These are dominant disorders, most of them, and or practically all. And in those cases, one mutation on one chromosome is enough to make the disease, so a dominant disease. If again, you happen to have one of the mutations previously reported to cause one of these diseases, okay, the diagnosis is fine, no biopsy is needed. The same is for the two confirmed cases. If we have these very disruptive so-called truncating mutations on both chromosomes, one from the father and one from the mother, a biopsy is not needed to confirm the diagnosis. But in all the other cases, and if there is uncertainty of the pathogenic role of one or both variants identified, then certainly the muscle biopsy is of very high value. And I will come back to that why. One question was, considering the spectrum of titin-related muscle diseases from congenital myopathy to limb-girdle, are these the same disease or very different diseases? Okay, this is a question of definition, but yes, practically they are very different. So we have titinopathies, all the range from those diseases that cause death already during pregnancy, before birth, and up to these very late onset mild foot drops that may show some symptoms only after age 70, and all in between. And they are very, very dissimilar, which can be easily identified on muscle MRI, where we have certain muscles more affected than others, and they can be totally opposite between the different forms of titinopathies. The next question is, will the treatment approaches be the same? No, probably not. So if something is going to cure titinopathies, the current understanding is that it has to go over some sort of genetic manipulation, but of course there is a long way to go. So the last question that was posted to me, if I have a known pathogenic variants, meaning genetic confirmation, is it of clinical significance if I have myopathy versus dystrophy? So this relates to the question above. These are terms. So if you have really confirmed titinopathy, meaning mutations that are fully clear to cause the disease, then you have titinopathy, which is usually a dystrophy, meaning some progressive loss of muscle tissue. There are, however, exceptions always to the rule, and we have some titinopathies that have mutations in regions of the gene which are active only in fetal life and are turned down after birth. In these cases, there is no progressive loss of muscle tissue, so the muscles that survived during fetal life will develop more or less normally. So basically, yes, they're all dystrophies, but there are some exceptions.
Thank you.
I think I can stop there, yeah?
Yeah, thank you. We have about 10 minutes left. So Anna, I'd like you to have some time, and then Sandra.
Did I use so much time?
No, we're fine. But
okay.
Thank you.
Thank you. Okay, I will just share my slides. I will probably jump a little bit because I don't want to take all the time left. So just need to share. Sorry, not this one.
While you're sharing,
sorry.
Do you want to answer the question from the chat as we're waiting?
exactly. Sandra, why don't you address that question about the reference sequences while Anna is sharing?
Sure. The reference sequences, I mean, that's one of the very complicated things about Titin. It's such a very, very big gene that we only really recently are starting to learn more about or have been able to describe. So there's a gene code, and then there's different blueprints, the different isoforms. So there's an isoform that's more specific for heart, and then there's an isoform more specific for muscle. And I'm not sure if this is more an American-based or limited to American labs, but in the genetic testing labs that we often use that issue the CLIA confirmed reports, they still use the cardiac isoform or the cardiac reference sequencing. So that's where sometimes there's a discrepancy, because in the muscle research setting, we use the muscle isoform. The good news that even though it looks very confusing, the report itself should say what reference sequence it's used. So anyone that knows a little bit about Titin would be able to remap it to the muscle-based isoform. I was in touch relatively recently with one of the bigger labs asking if at some point they would be willing to switch over using the muscle-based isoform, as especially now more and more research is being published and we're learning more about the new isoform, but that just seems to be a slower process to be adapted.
Well, in fact, there is a combined isoform with all of the exons, which doesn't really exist in reality, but allows one to number every single variant in relation to a protein sequence. And Björn, I believe many of us—
That's the preferred one.
Yeah, because that accounts for
also by Leiden database.
Yeah.
Yeah, so that's not adapted yet in the American CLIA labs at this time.
Right.
No, and that is a problem because based on that, and some labs leave out certain regions of the gene, so they do not even report if something is in those regions.
But as long as the reference sequence is numbered, it shouldn't be a problem for anybody in the community who's got questions. Any of the scientists and laboratories should be able to translate from one to another.
Yeah.
I think Dr. Sarkozy is ready.
Okay, I will just jump a few slides just for sake of time. And I think, as the title said, the main focus was this for understanding the natural history is why this is important for trial readiness. And I think because we are all aiming to identify possible cure at some point for titinopathies in particular, there's some natural history is one of the steps that is very important to be clarified. So clearly we need to understand more about genetic diagnosis for all the patients, clarify epidemiology, and most importantly, understand natural history and deep phenotypization, understanding all the phenotypic features of patients, and that include also MRI and muscle imaging for all patients. Then, of course, we need to have outcome measures, so under how to assess patients over time and identify changes over the years. And
so we're not yet seeing your slides. We're seeing just a blank screen. Have you selected presentation?
Yes, I am. Sorry, I have no idea why you can't see that.
In that case, just tell us in words.
No, I'm gonna try again. Can you see it now?
Yes,
Yep.
therapy.
Okay, all right, okay. So anyway, all of these steps, understanding natural history is one of the steps to identify possibly a right to trial readiness for these conditions. And so far, I don't know what's happening. It's not moving at all. So we understand the genetic diagnosis is extremely complicated, and we still have quite a lot of patients who are undiagnosed. For example, in our cohort of patients that we have in London, we have about 20% of the patients with Titin mutation, but only that are genetically diagnosed, but we have many more patients who remain undiagnosed because of the complexity of the genetic information, and probably it's much less rare, this condition, than what we think. So we really need to also create registries to make sure that patients are identified, and then we follow them over time to understand their progression and identify the clinical features of patients. So there have been everything is jumping here. So there have been quite a lot of studies, cross-sectional studies, to describe the features of patients. So we know a lot about onsets. A lot of patients, in particular with recessive titinopathy, are born with some features or present in early life. We know about the function, the difficulties of patients. We know the severity and the changes over time of the weakness. We know the frequency of respiratory involvement for the different type of titinopathies, and we know that there's a lot of forms of titinopathies. There is cardiac involvement. Now, these features might differ depending on the genetic background of titinopathy. So we know that this is different if you have a recessive early onset titinopathy, or if you have a late onset titinopathy, where frequencies and onset of problems may be different. So it's very important that we understand the differences between all these features, but we also understand how the progression, how the conditions progress over time, and understand the course and the evolution of the disease for each subtype of titinopathies. And this is what is still unknown. Now, we also need outcome measures, tools to assess these changes over time. And from other similar condition, early onset condition or late onset condition, we now have outcome measures to assess these changes. And these might be physio assessment, time test, goniometry to look at the progression of contractures in the joints or in the spine. We can look at motor function scales in young patients who are not able to walk yet, or in older patients who are ambulant or non-ambulant. We can assess the respiratory functions. All of these measures are very important, and we need to assess if these are valid also and useful to assess changes in patients with titinopathy in different ages and different phases of the disease. We can also use imaging to assess, and this in particular for Titin condition is very valid, varied tools. We can use ultrasound, which is not invasive and therefore is perfect for children or could not, for example, undergo a muscle MRI, but muscle MRI clearly is a very useful tool, and we can look at patterns of muscle involvement. In particular, we see that in titinopathy there are specific markers that have been recognized both in young children but also in adults with adult form, with in particular involvement in muscle, the semitendinosus, that is typically affected in these patients. But we also know that there are other muscles that are involved. So important we look at, this is not for Titin, but it's RYR1. It's important we look at the whole body. And there is the idea that we could use MRI as also a tool to assess progression over time and see how the muscles change from one year to the next, and use this to assess the validity of a treatment. So there are a lot of questions that are still open, and I think we need to understand how the condition changes over time, and if there are trajectories that we can identify, and which is the best outcome measures for specifically for this condition. So we need natural histories to answer all these questions. And we know that natural histories are valid tools to answer these questions, to identify the course of the disease and identify in particular the best measures. So there are two types of natural histories. There might be retrospective, when you look at patients that have already been assessed, and we often do that as the first step. When in particular, when it's difficult to put all patients together at the same time, so you look at historical data of patients that you already seen in clinic, or you can do prospective natural studies and assessing over time patients that are recruited in a study. So retrospective studies has been done for similar conditions, and we are also doing this for similar conditions in London and also in other centers in the UK, in the world. We have done that, for example, for Selenon-related myopathy, which is quite similar, and we are able to identify changes, but often retrospective studies are limited. You might be biased by the criteria of how you selected the patients. So really what you need is prospective natural history studies. Often, in particular for titinopathies, we see that changes might be small if you do studies for short term, and this has been shown for other similar myopathies like RYR1. So if you only study these conditions for short time, you might not be able to identify changes. And it's important that you study for a little bit longer and you use the right tools to identify change. So potentially some of these outcome measures could be the right ones, but might be needed to be assessed for multiple times over a longer time. And because these conditions are rare, it's very important that we pull together the efforts and don't do things only in single centers, but try to harmonize efforts and do it internationally. So we started on this path, and we have quite a large cohort of patients. So we're looking retrospectively and cross-sectionally. So we're trying to identify all the features that are relevant to help us to clarify natural history. We look at MRI. We try to improve the correlations between MRI and clinical features and onset and progression. And use also MRI and biopsy for clues for diagnosis. And I think this is also, supports what Professor Udd said already, that yes, so all of these tools are very important and often helpful in patients who don't have a clear mutation. And all of this to define better correlations between genotype and phenotype. But what we really would like to do, and this is a proposal that we are discussing now, also with charities involved in particular with patients who have central nuclear myopathy, which is one of the presentation of this, the recessive early onset forms, is to possibly set up a natural history. So we would like to start, if possible, a study, and we're trying to work on this to possibly do a study that could look into more detail into the natural history and look at children, because we assess children in London. So this would be what we would be focusing on, and try to understand what are the main functional tests that can identify changes over time, and also look and validate ultrasound and MRI as outcome measures, and possibly assess children on at least for 18 months, because as I said, 6 months would be too short. And possibly aiming for some of this could be some basic early data that could be then maybe replicated in larger studies, because as we said, these are rare diseases and one study might not be sufficient. So we really hope that all of this could be started at first maybe in London and then in other centers as well, to improve understanding and putting the basis to developing therapies. clearly, we hope that the similar studies could be also replicated in other centers and for other not only early onset titinopathies. And these are my contact details in case anybody's interested to hear about what we would like to do and how we would like to set it up. And clearly, this is not just my effort, but it's a joint effort for the whole team and together with my collaborators that are listed there. Thank you.
Great. Well,
Sorry for
thank you so much, Anna. So we're out of time, unfortunately. It's amazing how fast these sessions go. But Sarah, will you rejoin us if you want to say any last words? And I'll just say that many of us will be available in some of the chat rooms throughout the rest of today and tomorrow. And we invite everybody in the community to continue to reach out with questions, join registries, speak up, make your voices heard, and get involved.
Just want to thank everyone, all the attendees and all the speakers for today and for all your work. We're really grateful. And just remember that the lounge will be open today if people want to connect with people in the lounges. And then we have our program starting tomorrow. We're going to talk about clinical trial readiness. And I know Anna touched on some ways we get ready for clinical trials with natural history studies and outcome measures and things. And I just want to thank you so much for your presentations today. Oh, I could hear my son next door. He's clapping.
Thank you all.